All right, today we are diving into one of the most incredible scientific stories ever told. We're talking about the monumental quest to read, letter by letter, the entire genetic blueprint of a human being. You got it? The Human Genome Project. You know, it all kicked off with a question that sounds simple, but was just incredibly ambitious. Imagine this. What if we could read the entire instruction book for a human being? That one idea, right there, was the spark for a scientific journey that, honestly, changed everything.
Okay, so what are we actually looking at here? This is a human karyogram. The best way to think about it is like this. You see those 23 pairs of chromosomes? Those are basically the chapters in our biological instruction book. And packed inside each one is that famous, tightly coiled DNA that holds all our genetic secrets. So, how big is this book we're talking about? Get ready for this number. 3.1 billion. That's how many letters, or base pairs, make up our DNA. And the mission? To read every single one in the right order. The scale of that task is it's absolutely mind-boggling.
Okay, so you've got this book with over 3 billion letters. Where do you even start? I mean, how could anyone possibly tackle that? Well, it was clear right away that this wasn't a job for one person or even one lab. It was going to take a whole new way of doing science on a global scale.
And so, the first real push to do this came in the form of this incredibly audacious public project. Think of it like a grand alliance for science, pulling in the best and brightest researchers from all over the world.
You can actually get a sense of just how big this was by looking at the official logo. See, this wasn't just a biology project, not by a long shot. It was this huge, multidisciplinary thing that pulled in physics, chemistry, computer science, and really importantly, ethics.
So, let's break down the game plan for this public effort, the HGP. It kicked off in 1990 with a pretty ambitious goal: sequence the whole thing in 15 years. The budget? Around $3 billion. It was this huge international team-up, with 20 different universities and centers involved. Their method was slow but super reliable. But here's the most important part, the real philosophy behind it all: open science. Every single day, they released all their data to the public, for free, for everyone. But then, just as this massive public project was chugging along, about 8 years in, the whole story gets a massive injection of drama. A private challenger steps into the ring, and suddenly, this scientific quest turns into a high-stakes race.
And that challenger? A scientist named Craig Venter. In 1998, he launched his own company, Celera Genomics, and he made this incredibly bold promise. He said they could sequence the entire genome faster, years ahead of the public project, and way cheaper too.
So now you've got these two teams, and they couldn't be more different. We're talking a total clash in both method and philosophy. The public HGP was slow and steady, map first, then sequence. Celera, on the other hand, used this wild whole genome shotgun method. Basically blow it all into tiny pieces, sequence them all at once, and then use massive supercomputers to stitch it back together. But the real difference, the fundamental one, was about the data. The HGP's work was a public resource. Celera's goal? To patent genes and sell access to their database.
And just like that, the race was on. This was more than just science. It was a clash between two totally different ideas about what the future of science and business should look like.
This timeline really shows you how fast everything escalated. The public project starts its 15-year marathon in 1990. Then, boom, 1998. Celera jumps in and turns it into a sprint. Things get really intense until March 2000, when a huge political statement just changes the entire game. And that leads to a surprise joint announcement just a few months later. All that competition, it actually pushed the public project to finish and declare victory in April 2003, a whole 2 years early.
Now this right here, this was the game-changing moment. In March 2000, you have the President of the United States, Bill Clinton, and the Prime Minister of the UK, Tony Blair, releasing a joint statement. And they said that all researchers should have unencumbered access to the genome. It was a massive public endorsement of the HGP's open science approach.
And the fallout from that statement was, well, it was immediate and it was huge. Wall Street basically freaked out. They saw it as a direct attack on Celera's whole business model, selling the data. The biotech sector lost an estimated $50 billion in market value in just 2 days. Unbelievable.
So with all this political and financial pressure, the race didn't really end with one person breaking the tape. It was much weirder than that. It ended in a truce, a really surprising one. In June of 2000, you have the leaders of both projects, the public one and the private one, standing side by side at the White House, announcing that they'd both finished a working draft of the genome.
But here's the thing. The race to just get the sequence, that was really just the beginning. The real story, the part that matters to us today, is the legacy. So, what did we actually learn from reading our own instruction book?
And right off the bat, some of the discoveries were just humbling. For years, scientists thought we must have over 100,000 genes. I mean, we're complex beings, right? More complexity must mean more genes. Well, the real number? It's closer to 22,300. That was a shock. It told us that our complexity doesn't come from the number of parts, but from how we use them. We also found out that our genome is full of these weird repeating chunks of DNA, and that most of our protein families aren't even unique to us vertebrates.
If you really want to get a sense of just how much information we're talking about, just look at this. This is the Human Genome Sequence, printed out and bound in books. It's an entire library, a library of life. And for the first time in history, the doors were finally open.
And the impact this has had on our world since then? It's been absolutely transformative. In medicine, it's led to things like targeted cancer therapies and new ways to diagnose diseases like cystic fibrosis. In forensics, well, it completely changed the game with DNA evidence. And for researchers, it blew open brand new fields of study in everything from human evolution to making better crops.
You know, what's really incredible is that the people running the project, they saw the future. They knew this powerful knowledge would come with some really tough questions. So they did something that was basically unheard of for a big science project. Right from the start, they set aside 5% of their entire budget just to study the ethical, legal, and social implications. They called it the ELSI program.
But wait, there's one last twist in this story. That big mission accomplished announcement back in 2003, well, it wasn't quite the whole picture. They had sequenced about 92% of the genome, but the really tricky, repetitive parts were still full of gaps. It actually took almost 2 more decades of incredible new technology to get the first, truly complete, no gaps allowed sequence in 2022. And the final piece of the puzzle, the Y chromosome, wasn't fully finished until 2023.
So there you have it. After a journey that spanned more than 30 years, we finally have it. The complete, high-quality map of ourselves. It is, without a doubt, one of the greatest scientific achievements in human history. And that leaves us with the biggest question of all. We have the map, so where do we go next?